Okay, guys, it's our last day together. Not really. We'll see me on the 20th. And if you're taking cell biology also, you'll see me twice on the 20th, because apparently I'm proctoring your exam. Just saying. Okay. So this is the last lecture, and it's really just like a little glimpse at cool things that scientists get to study. And then after this is done, you can grab your exams and, we could do a quick review if there are questions that people have. If not, I don't know if it's too early, it's still a little ways from the exam. But if you have questions, we can talk for a minute. If not, feel free to use the time to come up and talk to me. Okay. Any questions about anything? Yes?
Will you have office hours this week still?
I have office hours today. And if you need to see me, email me, and we'll find a time. Yes?
When do you think we'll have our grades for the projects?
Definitely before the final exam. Sometime the next day or two, I'm hoping. Don't hold me to that. Any other questions? Good. Okay, so final exams May 20th. Exams pick up after class or in my office. I did bring a copy of the exam if you want to actually quickly look at what you got wrong. Okay, so this is what you need to worry about for the final if you are taking it. So all the translation stuff we did, from binding the correct amino acid to the correct tRNA, to the actual mechanism of translation, initiation, elongation, termination, DNA damage. It's really annoying. Repair, the different mechanisms of repair, double-strand break repair, the DNA damage response, so the recruitment of repair proteins, and how the cell cycle is halted during damage, in presence of damage. Then we just spent a short time talking about retroelements. I should have said retroelements is actually only two out of three categories. We'll say mobile genetic elements would be more appropriate. And today we'll talk very briefly about control of retroelements by the APOBEC proteins. So to understand that, you need to understand a little bit about control of HIV by APOBEC 3G. So APOBEC 3G is a cytidine deaminase, and what happens is it is packaged, so you've got one cell here, and you've got another cell here. So this cell's already infected, HIV's gone through its whole life cycle has made new virus and is going to bud out and then go and infect the next cell. And before it leaves to infect the next cell, it packages APOBEC 3G and 3F. And then 3G and 3F go on in the next cell during reverse transcription, where you have single-stranded DNA. You get deamination of Cs to Us, which results in, after DNA synthesis, hypermutation of the viral genome. And if you have a hypermutated genome, do you think you're going to be a very effective virus? Monday. Okay, yeah, you guys are totally dead people today. Wake up. This is cool stuff, okay? Cool stuff. Woohoo! Okay, thank you. Someone's gotta be awake. This is the cool stuff. This is why you have been studying all this stuff, so you can go do cool research. And so I'm trying to give you a little glimpse at cool research. Okay, so this is kind of amazing that there's this protein that is in cells that likes to bind to viral things and get incorporated into baby virus, and is basically a time bomb for the virus. As soon as that virus goes into the next cell, it hypermutates the DNA, and then the infectious virus that's produced is like nil. It's like none. So what question does this beg? Yes? Why is HIV still a problem? Great. Perfect. It's like I planned it. Yeah. Right? So you should be asking, wait a minute, if we've got this, and by the way, this is interferon induced, so you guys should have all seen it on your gene list, and there's actually one group that did their project on this. So remember, antiviral activity results in interferon production, which results in gene expression, and one of the genes that's induced is ABOBEC3G and probably 3F also. So this is an innate antiviral response. And yes, why do we still get HIV? That's because of this round circle here. That's green, it says VIF. And that's a viral protein that has evolved to counteract the activity of ABOBEC3G. So again, ABOBEC3G is a cytidine deaminase, so it deaminates and removes this amine group from cytosines, and you end up with uridine or uracil, and you end up with, so this happens during reverse transcription. So HIV has a RNA genome, which is reverse transcribed to DNA, and the first step has a single-strand DNA molecule, and that's what's deaminated. And then when you complete making double-stranded DNA, you end up with a genome that has lots of G to A hypermutation. So what does HIV do? HIV expresses this amazing protein called VIF. And VIF, it stands for virion infectivity factor. And in the beginning, when VIF was first discovered, all people knew was that VIF, if you have VIF, then you have infectious virus. Without VIF, you don't have infectious virus. That was the only thing that they knew. And then someone finally figured out why. Because they're comparing two different cell types that were identical, except for the difference was one cell type could easily be infected and the other one couldn't. And they found out the one difference between these cell types were these ABOBEC proteins, specifically ABOBEC3G. So it's kind of amazing. So VIF actually binds colon-5 ubiquitin ligase and forces polyubiquitination of ABOBEC3G, and it's degraded. Brilliant, right? You're a virus. What better to do than throw the immune response into the garbage, right? Brilliant. Why am I talking about this? So I did my PhD on this, and I went to many conferences, and over time, people started, HIV field is insane, and you end up with 20 million people working on something, so everything goes really fast. And within like a 3-year period, the whole mechanism of APOBEC and everything was worked out, and the mechanism of HIV inhibition of APOBEC was worked out, and people were like, oh no, what do we do next? And people started thinking about, well, where does APOBEC come from? Why do we have APOBEC? It must have evolved from somewhere, and then there's this whole story that came from there. And so that brings me to mobile genetic elements, which we spoke about the other day. And so some things that I want you to think about, and the jury's still out on how, well, we'll get back to this. So remember of the mobile genetic elements, retrotransposons, so those are the ones that have a reverse transcription step, those make up a huge part of our genome. I think we say LTR, ALU. I mean, I think it's supposed to be like 30% or something like that. But many of them are no longer active in humans. So there's all these jumping DNA sequences that have been great because they've basically been responsible for the diversity in our genome, and probably the evolution to be I don't know, Homo sapiens. If you look, I think there are some people who are really into thinking about evolution of things. There's some really great papers looking at the evolution of the APOBEC proteins all the way from fish, and like where the different proteins diverge. And actually, the APOBEC proteins themselves are only in mammals, but there are related proteins that go all the way back. So anyways, so a whole bunch of these are not active. So if you look at this, these are two different retrotransposons, and they're very, very present. So some of them are called endogenous retroviruses, and they actually form a virus, but it stays in the cytoplasm and then just goes back into the nucleus, and that's the cycle. And some of them just kind of jump around within the nucleus. And you can see they're very, very common in the genome. Some of them in mouse have up to 200,000 copies. Human endogenous retroviruses have 230,000 copies. Then there's the L1, which are these lines that litter our genome. And there you see in human 850,000 copies in our genomes. They're very present. But if you look at the number of active retro elements, they're very, very low. And there was a approximation in mouse of MUSD, and less than 10 were found that are active. And with L1, in mouse there's a lot more, but in human, from 850,000 that are present, it's only about 80 that are active. And this is because there's been an acquisition of a lot of mutations in the retro element genomes. Okay? And so this is why we're talking about it, because what do the ApoBec proteins do? They hypermutate. And it's believed that they may have arisen from, basically they were able to control these retro elements in our genomes because too much transposon, too much transposition is going to be harmful, right? So if you've got all this DNA jumping around everywhere, there's a certain point where it increases diversity, and that's good for survival of a species. But if you hypermutate like crazy, well, not hypermutate, but you move around too much, you're going to disrupt lots of genes, inappropriately activate different genes. So there has to be a way to control the activity of these mobile genetic elements. Is the logic there? You guys following? Okay. So in 2005, there was a really great paper where people they actually sequenced two different retrotransposons, IAP and MUSD, in the presence and absence of ApoBec proteins. And they looked at ApoBec3G and mouse A3. So if you compare no ApoBec to plus ApoBec, and mind you, this is done in vitro, so it's very artificial. They're really just adding the transposons they're adding the ApoBec proteins in the presence and absence with the transposons, and they're looking to see the mutations. And each little red line shows a mutation, a G to A mutation. And so you can see they're fairly hypermutated in the presence of the ApoBec proteins, but where there's no ApoBec proteins, you see no mutation, suggesting that these ApoBec proteins may have a significant role in hypermutation of these retro elements and have been involved in decreasing the activity of retrotransposons through hypermutation. Okay? That's kind of awesome, isn't it? We're like, what? 56 Can you calculate my grade for me? No! It's going to be on Blackboard. Don't worry. You're going to be okay. Okay. So, if you want to learn more about this, actually, before I end, does anyone have any questions about this concept? It will be on the final. This isn't just for fun. So make sure you think about this and understand it. And yes, I'm recording this so you can listen to it later. And then I'm also giving you this talk. This is by this guy who was there when this was all happening, and he's very cool. He does a nice long talk about ApoBec proteins, and so you can go watch it. Also, I think I put a lot of the citations. Yeah, they're all really great reviews to read about this. It's just a very cool area that people study. Okay, yes.
So, HVAC proteins are only active in presence of viruses.
ABOBAC proteins are induced by the antiviral response. So they're going to be more active in the presence of viruses because that's what's induced their expression. But they might have other. So what I didn't tell you is that, do I have a whole, no, I didn't put the families in. What I didn't tell you is that they arose from two related genes, one of which is, the reason it's called APOBAC is because it's called apolipoprotein B catalytic editing, something or other. So basically what it does is it edits apolipoprotein B so that it can form two different proteins. It forms a premature stop codon cell. In certain conditions, you get one protein. In certain conditions, you get another. Nothing to do with the immune response, right? It has to do with lipid metabolism, right? And then it's related to another gene called AID, which is involved in somatic hypermutation, so generating antibody diversity. So those are also, I guess that would kind of be induced by interferon also, right? But these tend to be active. Well, there could be things that people haven't found yet that are substrates for editing. So. Any other questions? That's a good one. I know it's the end of the semester. It's hard. It's hard. Okay. Well, then that's it for this. So we could do a quick review session if anyone wants. If you don't want to, you can run away. If you want to get your exams.